Chapter 7: Hormonal Signaling and Membrane State. Hormonal signaling is constrained by the membrane that receives, organizes, and transmits the signal. Hormones arrive as inputs, but the output they generate depends on how receptors are positioned, how they move, and how they interact with downstream machinery within the lipid environment. The membrane defines these conditions by controlling packing density, domain formation, and electrical stability. When structure supports alignment, hormonal signals produce coordinated responses. When structure is altered, the same hormone produces a different output because the context that governs receptor behavior has shifted. Receptors are not isolated sensors. They are embedded components whose activity depends on their surrounding lipids. Membrane composition determines receptor conformation, influencing how easily it transitions between inactive and active states. In tightly organized regions, receptors cluster and interact with coreceptors and signaling proteins in a controlled manner. Ligand binding leads to coordinated activation across multiple receptors, producing a signal that can propagate efficiently. When the membrane lacks this organization, receptors remain dispersed and activation events fail to combine into a unified output. The hormone is present, but the signal does not scale because the structure does not support amplification. Mobility within the membrane sets responsiveness. Receptors must move laterally to encounter partners and form signaling complexes. Adequate mobility allows rapid assembly when a hormone binds, producing a timely response. Excessive mobility prevents stable interactions, causing complexes to dissolve before the signal can propagate. Restricted mobility delays assembly, reducing sensitivity to changes in hormone concentration. The membrane defines this balance, determining how quickly and how reliably hormonal signals are processed. Domain organization directs specificity. Different hormones rely on distinct receptor systems that must remain separated to avoid interference. Lipid domains create these separations by concentrating compatible components and excluding others. This ensures that each signaling pathway operates within its own defined region. When domains are stable, signals remain specific, and crosstalk is minimized. When domains fragment, components from different pathways mix, leading to overlapping or conflicting signals. The system becomes less precise because the structural boundaries that enforce separation have degraded. Electrical properties influence receptor activation and downstream signaling. Many hormone receptors and associated channels respond to changes in membrane potential. A stable electrical environment supports predictable activation thresholds and consistent signaling dynamics. When permeability increases and potential fluctuates, these thresholds shift. Receptors may activate at lower or higher input levels, and signaling duration may extend or shorten unpredictably. Ion fluxes that accompany these changes further influence intracellular pathways, integrating electrical instability with biochemical signaling. Hormone sensitivity is therefore not fixed. It emerges from the interaction between hormone concentration and membrane state. A well-structured membrane can produce strong responses to modest inputs because receptors are positioned and organized to amplify the signal. A disorganized membrane requires higher input to achieve the same effect and may still produce inconsistent output. This alters how the system interprets hormonal signals, affecting processes that depend on precise regulation. Termination of hormonal signaling depends on the same structural conditions. Receptors must disengage, complexes must disassemble, and downstream pathways must reset. This requires coordinated access of termination mechanisms within the membrane. When structure supports this coordination, signals end cleanly. When it does not, activation persists or overlaps with new inputs, increasing variability in response. Hormonal signaling is therefore an expression of membrane state. The hormone provides the input, but the membrane determines how that input is translated into output. Changes in lipid composition, domain stability, and permeability alter the behavior of receptors and the pathways they control. The system responds not only to the presence of hormones, but to the structural conditions that define how those hormones are processed. Receptor sensitivity as a membrane-dependent property. Receptor sensitivity is set by the physical environment in which the receptor is embedded, not by receptor number alone. The membrane determines how easily a receptor transitions into its active state, how long that state is maintained, and how effectively it engages downstream partners. Lipid packing, domain organization, and local electrical conditions define the baseline from which a receptor responds to input. Change those conditions, and the same receptor population produces a different response to the same hormone concentration. Conformation depends on surrounding lipids. Receptors span the bilayer and interact directly with lipid head groups and acyl chains. These interactions influence the shape of the receptor and the stability of its active and inactive states. In a tightly packed environment, the receptor is constrained, and activation requires sufficient input to overcome that constraint. Once activated, the same environment can stabilize the active conformation if clustering occurs within a compatible domain. In a more disordered membrane, the receptor experiences less constraint, lowering the threshold for activation but also reducing stability. Activation becomes easier but less controlled, and signals are more likely to fluctuate. Clustering amplifies sensitivity by increasing local receptor density. When receptors are organized within domains, ligand binding to one receptor increases the likelihood that neighboring receptors will also engage. This produces a coordinated response that scales with input. The membrane enables this by supporting lateral movement within defined regions and by concentrating compatible proteins. When domains are disrupted, receptors remain dispersed, and activation events do not combine. Sensitivity decreases because higher input is required to achieve the same level of coordinated activation. Coupling to downstream pathways depends on proximity and alignment. Receptors must interact with specific proteins to transmit a signal. The membrane positions these components so that interaction occurs rapidly after activation. If the membrane maintains this organization, signaling proceeds efficiently. If components are misaligned due to changes in lipid composition or domain structure, the receptor may activate without successfully engaging downstream machinery. This creates a state where binding occurs, but signal transmission is weak or incomplete. Electrical conditions modify sensitivity further. Changes in membrane potential influence the behavior of voltage-sensitive receptors and associated channels. Stable potential supports consistent activation thresholds, allowing predictable responses to input. Fluctuating potential shifts these thresholds, altering how receptors respond to the same signal. Ion fluxes that accompany these changes feed back into receptor behavior, reinforcing or dampening sensitivity depending on the direction of change. Desensitization and resensitization are also membrane-dependent processes. After activation, receptors must be reset to respond again. This involves disassembly of signaling complexes, internalization, or conformational changes that return the receptor to its baseline state. The membrane controls how quickly and efficiently this occurs by governing access of regulatory proteins and the mobility required for receptor trafficking. When structure supports coordinated disassembly, receptors reset cleanly. When structure is compromised, receptors may remain partially active or fail to return to a responsive state, reducing overall sensitivity. Receptor sensitivity emerges from the interaction between receptor structure and membrane environment. It reflects how the membrane constrains movement, organizes interactions, and maintains electrical and chemical stability. Adjustments to membrane composition change these conditions, altering how receptors interpret and respond to signals. The system does not rely solely on receptor abundance to regulate sensitivity. It relies on the structural context that determines how those receptors behave under varying inputs. Insulin signaling and membrane composition. Insulin signaling begins at the membrane, but is determined by whether that membrane can organize and stabilize the insulin receptor and its downstream machinery under load. The insulin receptor is a transmembrane tyrosine kinase that requires precise positioning and clustering to transition from ligand binding to effective signal propagation. The lipid environment defines how easily this transition occurs. Packing density, domain formation, and local electrical conditions set the threshold for activation and the efficiency of downstream coupling. When the membrane supports alignment, insulin binding produces a coordinated cascade that moves glucose transporters to the surface and shifts cellular metabolism. When it does not, binding occurs, but signal transmission is incomplete or delayed. Receptor activation depends on proximity and orientation. Insulin binding promotes autophosphorylation of the receptor, which requires that receptor subunits are correctly aligned within the membrane. Lipid composition influences this alignment by controlling how the receptor sits within the bilayer and how it interacts with neighboring receptors. In membranes that support domain formation, receptors are concentrated into regions where interaction probability is high. This increases the likelihood that activation will propagate beyond the initial binding event. In membranes that lack this organization, receptors remain dispersed and activation remains localized, reducing the strength of the signal. Downstream signaling requires rapid recruitment of adapter proteins and kinases to the receptor. These components must encounter the activated receptor within a defined time window to propagate the signal. The membrane governs this process by controlling lateral mobility and spatial confinement. When mobility is balanced with stability, adapter proteins are recruited efficiently, and the signal proceeds through the pathway without delay. If mobility is excessive, components fail to remain in contact long enough to transmit the signal. If mobility is restricted, recruitment is slowed, and the signal weakens before it can be amplified. Membrane composition also influences the formation of lipid domains that support insulin signaling. Certain lipids promote environments where signaling complexes can assemble and remain stable long enough to complete their function. These domains act as platforms where the receptor, its substrates, and downstream enzymes are co-localized. When these platforms are intact, signaling proceeds with precision. When they are disrupted, components disperse, and the pathway becomes less efficient, requiring higher insulin levels to achieve the same effect. Glucose transporter translocation depends on this structural framework. After activation, signaling pathways direct vesicles containing glucose transporters to the membrane. Fusion of these vesicles requires membrane regions that can accommodate curvature and maintain integrity during the process. Lipid composition determines whether these regions form and whether fusion occurs efficiently. If the membrane lacks the necessary properties, transporter insertion is reduced, limiting glucose uptake even when upstream signaling is partially active. Electrical and ionic conditions further modify insulin signaling. Changes in membrane potential and ion gradients influence the activity of kinases and other signaling proteins involved in the pathway. Stable conditions support consistent signaling, while fluctuations introduce variability in activation and propagation. Increased permeability or disrupted gradients alter how these proteins behave, contributing to reduced signaling efficiency. Insulin resistance emerges when membrane conditions no longer support coordinated signaling. The receptor may still bind insulin, but clustering is reduced, adapter recruitment is impaired, and transporter translocation is inefficient. The system compensates by increasing insulin levels, but this does not restore the structural conditions required for efficient signaling. The outcome is reduced responsiveness despite the presence of adequate or elevated input. Membrane composition therefore determines how insulin signaling is executed. It sets the conditions for receptor activation, pathway propagation, and final functional output. Changes in lipid organization alter each stage of the process, shifting the system from coordinated response to partial or inconsistent signaling because the structural framework required for efficient communication is no longer maintained. Steroid hormone interaction with lipid environments. Steroid hormones do not approach the cell as purely external signals. Their structure allows them to partition directly into the lipid phase, making the membrane both an entry point and an active participant in how the signal is processed. Before a receptor is engaged, the hormone has already interacted with the bilayer, and that interaction is shaped by lipid composition, packing density, and domain organization. The membrane determines how readily the molecule inserts, how it distributes within the bilayer, and how efficiently it reaches its intracellular targets. Partitioning depends on compatibility with the lipid environment. Steroid molecules align with the hydrophobic core of the membrane, positioning themselves among fatty acid chains. In membranes with tighter packing, insertion is more constrained and movement through the bilayer is slower but more controlled. In more disordered membranes, insertion occurs with less resistance, but distribution becomes less predictable, and the hormone may diffuse unevenly across regions. This affects how uniformly receptors are engaged and how consistent the resulting signal will be across the cell. Movement through the membrane is not uniform. Lipid domains create regions where diffusion rates differ, altering the path a steroid hormone takes as it traverses the bilayer. Order domains can slow movement and concentrate the molecule in specific regions, increasing the likelihood of interaction with localized receptors or transport mechanisms. Less ordered regions allow faster diffusion but reduce the probability of sustained interaction with any single receptor system. The membrane therefore guides the hormone towards certain signaling routes while limiting others based on its internal organization. Receptor engagement depends on how the hormone exits the membrane and encounters its binding site. Many steroid hormone receptors are located within the cytosol or nucleus, requiring the hormone to cross the bilayer and enter the intracellular environment. The efficiency of this transition is influenced by membrane permeability and the stability of the lipid barrier. If the membrane is too rigid, passage slows, delaying signal initiation. If it is too permeable, the hormone may diffuse rapidly but without controlled distribution, leading to uneven receptor activation. Membrane composition also affects the availability and behavior of membrane-associated steroid receptors. Some receptors are embedded within or closely associated with the membrane, responding to hormones that remain within the lipid phase. These receptors depend on local lipid conditions to maintain their conformation and interaction with signaling partners. Changes in lipid composition alter how these receptors function, shifting their sensitivity and the pathways they activate. Steroid hormones can modify the membrane as they pass through it. Their presence within the bilayer alters local packing and can influence the behavior of nearby lipids and proteins. This creates a feedback effect where the hormone not only responds to the membrane environment but also contributes to changes in that environment. These changes may be transient, but they can influence how subsequent signaling events unfold. Electrical and chemical conditions intersect with these interactions. Changes in membrane potential and ion gradients can influence how steroid hormones partition and move, as well as how receptors respond once engaged. Stable conditions support predictable signaling, while fluctuations introduce variability in both hormone distribution and receptor activation. The interaction between steroid hormones and lipid environments therefore precedes and shapes the signaling process. The membrane determines how the hormone enters, where it travels, and how effectively it engages its targets. Changes in lipid composition alter each of these steps, producing different outcomes from a same hormonal input because the structural medium through which the signal passes has been modified. Hormonal instability as structural drift. Hormonal instability reflects a gradual loss of membrane conditions that support consistent receptor behavior, signal propagation, and termination. The system does not fail because hormones disappear or receptors vanish. It becomes unstable because the lipid environment that governs how signals are processed drifts away from the state required for coordinated function. This drift alters thresholds, timing, and amplification, producing variable outputs from the same hormonal input. The process begins with changes in lipid composition that affect packing and domain formation. As membranes shift toward less stable configurations, receptor positioning becomes less consistent. Clusters that once formed reliably now assemble intermittently or not at all. This changes the relationship between hormone concentration and response. At times, small inputs produce exaggerated effects when clusters form unexpectedly. At other times, larger inputs fail to generate sufficient activation because receptors remain dispersed. The signal becomes inconsistent because the structural conditions that define sensitivity are no longer stable. Mobility within the membrane contributes to this variability. Receptors must move to interact with coreceptors and downstream proteins. When lipid organization drifts, this movement becomes either excessive or restricted depending on local conditions. Excessive movement prevents stable complex formation, shortening signal duration and reducing amplification. Restricted movement delays interaction, increasing the time required for activation and reducing responsiveness. The system shifts between these states as membrane properties fluctuate, producing irregular signaling patterns. Domain instability disrupts pathway separation. Hormonal systems rely on distinct regions of the membrane to maintain specificity and prevent interference between pathways. As domains lose definition, components from different signaling systems begin to overlap. This creates crosstalk where signals intended for one pathway influence another. The result is a loss of clarity, where outputs reflect mixed inputs rather than discrete signaling events. Hormonal regulation becomes less precise because structural boundaries that enforce separation are weakened. Electrical instability reinforces these effects. Changes in membrane permeability alter ion gradients and membrane potential, shifting activation thresholds for receptors and associated channels. Fluctuating potential changes how receptors respond to the same hormone level, producing variability in both onset and duration of signaling. Ion fluxes that accompany these changes influence intracellular pathways, adding another layer of inconsistency to the system. Termination processes are affected in parallel. Receptors must disengage, complexes must disassemble, and signaling molecules must be cleared to reset the system. Structural drift interferes with these processes by dispersing termination machinery or altering the conditions required for deactivation. Signals persist longer than intended or fail to terminate cleanly, overlapping with new inputs. This accumulation of partially resolved signals contributes to instability, as the system never fully returns to baseline. Transport and distribution of hormones are also influenced by membrane state. For hormones that interact with or pass through the membrane, changes in lipid composition alter how they partition and move. This affects how uniformly receptors are engaged across the cell. Uneven distribution leads to localized differences in signaling, further increasing variability in response. Hormonal instability, therefore, emerges from a membrane that no longer enforces consistent conditions for signaling. The same inputs are present, but the system processes them differently over time because the structural framework that governs receptor behavior, interaction, and termination is in flux. Restoring stability requires restoring membrane composition and organization so that receptor systems operate within defined boundaries, allowing signals to be generated, propagated, and resolved with consistency rather than variability.